have the pleasure of introducing to you Mia, who will talk to us first. She's a master's student here, grew up local in Rancho, loving the deserts and the mountains and the coast, and that passion has kind of carried her with a bend towards plants and conservation of rare plants increasingly. We first had her at the garden as an intern, and then she went and finished her studies at Cal Poly San Luis Obispo, had some internships, some jobs working for national parks in Southern California and the Huntington, and we're very fortunate to have her back doing some very exciting work out in our deserts, focusing on a rare member of the mint family. And so thank you, Mia. Let's hear about that.
Thank you so much for that introduction. I'm so excited to be here talking to you all today about my master's thesis research that I've been working on for the past year. That my title is an integrative study examining the disjunct distribution of Salvia funerea Death Valley sage. And so I want to start us off with some background on my study species. Salvia funerea is a drought deciduous shrub that is an endemic of the Mojave Desert, and it is restricted to carbonate, limestone, and dolomite mostly soils, and it has these fun spine-tipped leaves, and it's also very, very hairy. Its calyces are so densely hairy, they look like little cotton balls, and they're really, really cute. And then it has these stunning deep violet flowers that really pop in the desert. And it really likes to live on these super rocky slopes and outcrops. And so if you really like rock scrambling, please come do field work with me this year. I promise it'll be a great time. And then the most recent phylogenetic studies have placed this species within Salvia subgenus Audibertia, which includes all 19 species of Salvia that occur in California, Baja California, and then further into section Echinospacey, which includes some other furry, spiky salvias, including Salvia cartuacea, thistle sage, Salvia californica, which is restricted to Baja California, and Salvia gradii, which is a rare member in the Sonoran Desert near the Salton Sea. So, a cool group, all desert adapted species. And like my title promised, Salvia funerea has a super interesting disjunct distribution in the Mojave Desert. All the green points on this map came from georeferenced herbarium specimens and research grade iNaturalist points. And so you can see there's a much larger group of occurrences in the northern region of the range, inside of Death Valley National Park, and then right outside on adjacent BLM lands in the Shoshone area. And there's 4 known occurrences across the Nevada border in the Last Chance Range. And then 80 miles away, south of I-40 near Amboy, California, there's a much smaller group of occurrences in 3 mountain ranges, the Bristols, the Marbles, and the Ships. And so because of this limited distribution, California Native Plant Society has given this species a rare plant rank of 4.3, which means it's rare, but it's not super rare. And it's also not currently considered to be super threatened because there are a decent amount of occurrences and it's pretty well protected by the national park. And then NatureServe has also given it a rank of G4 globally, or apparently secure. And then it has the same subnational rank in the state of California, apparently secure. But in Nevada, it has a rank of S1, which means it's considered critically imperiled because there's only 4 known occurrences in Nevada. And so I have 3 main objectives for my master's thesis. First, I want to estimate gene flow and genetic diversity within and between populations throughout the range of the species in between those northern and southern regions. I want to investigate climatic and morphological differences across the species range, and again, between the north and the south. And then I want to use that information to update conservation status of the species and make recommendations for any future conservation actions that it might need, because it is a resident of the Mojave Desert, which we all know is an area that is being heavily impacted by climate change. And so my study is integrative, and then I'm combining several different research methods in order to more fully meet those objectives that I just laid out for you. And so I'm doing extensive fieldwork throughout the range of the species, collecting genetic samples and voucher specimens, and taking detailed field notes on abundance and plant health condition and habitat characteristics and all those good things. I'm also doing some morphometric analyses to look for any possible morphological differences between plants in the south and plants in the north. I'm doing some species distribution modeling to look at habitat suitability, to maybe identify some new areas to look for the species that aren't currently recorded, and also to make predictions on how the species range might change under future climate scenarios. And then finally, I'm doing my population genomic analyses to look at genetic diversity and population structure throughout the range. And so this last spring, I got to do a lot of field work. I think I did like 8 total weekend trips during the semester. And so each of these polygons on this big map, and there's 17 of them, they are my target localities for genetic sampling, and they are delineated by mountain range. So each polygon represents a different mountain range where Salvia funerea occurs. If we zoom in on the north, there's 14 polygons throughout Death Valley National Park and on adjacent BLM land. That X is a polygon that used to be there, but it turns out all the points that were in that mountain range were georeferenced wrong, and they're actually in a different mountain range. So that polygon doesn't exist anymore. So we're ignoring it. Didn't have time to update my map, sorry. And then in the south, there are 3 polygons representing those 3 mountain ranges that Salvia funerea is known from. We've got the Bristol Mountains here, the Marble Mountains here, and then the Ship Mountains down here. And so far, I have been able to sample from 16 of my 17 target localities. I've actually been to the last one, up in gray in the Last Chance Range of California. This species is fun and occurs in the Last Chance Range of California and the Last Chance Range of Nevada. Fun fact. And I went the hard way. Chloe was with me, and it ended up being a 6-hour off-road adventure that we didn't know we were getting into that day. And so that was not a successful sampling trip, but I will go back the easier way this coming spring to sample from there. And then my other caveat is that I've been to the Marble Mountains down in the south, but I was only able to find 3 plants and sample from those 3 plants. So I have to go back again in the spring to find some more to sample from. Some highlights from my fieldwork so far are seeing quite a few floral visitors. So we've got this really cute little native bee that I saw in Cottonwood Canyon in Death Valley National Park when I was there with Matthew. And I've gotten to see several other native bees and bee flies and cool insects on these plants. That's really exciting. And then just like an interesting observation, there's this one area in the Bristol Mountains where I saw this interesting like lighter purple flower color. Usually they're this dark purple, and those plants were not happy. So there might be some correlation with drought stress in the area, maybe having something to do with that different flower color. Interesting. So I've also been able to do some morphometric studies. I measured 31 herbarium specimens here in our herbarium, CalBG, and I measured 7 different floral and leaf traits. And then I conducted t-tests and a principal components analysis to look for any significant differences in those traits between specimens collected in the north and specimens collected in the south. And I found no difference. So all of my p-values for my t-tests are really big. And my PCA is just sort of a cloud. There's no separation between those two groups, which makes sense. That's also what I've been seeing in the field. No noticeable differences between those two areas. I also did some preliminary climate analysis exploring variables that I maybe want to include in my species distribution model. And for this, I downloaded 7 climate variables from the PRISM 30-year normals dataset. And then I did the same t-test and principal components analysis to look for differences between the North and the South. And for this one, I did find significant differences in all of my variables except for precipitation. And you can see there's a lot more data for the North. It's a much bigger area than the South, so that's a consideration when looking at those results. And I actually don't think I'm going to use this data set in my species distribution models at all. I think I'm going to use world climate variables instead. So update on that soon. Then this summer, I was in the lab extracting DNA from all 253 samples of Salvia funerea that I collected in the spring, as well as 4 outgroups. And I did that using a modified CTAB protocol, pretty standard. And so far, I have been able to submit 2 libraries of 96 samples each for sequencing. And I'm doing ddRAD sequencing, which basically chops up the genome into lots and lots of little pieces so that we can sequence loci from all across the genome, many loci all across the genome, to then align and look at genetic diversity through differences. And I've gotten the results back from one of those sequencing runs. And so I have some very preliminary genetic results to share with you all. And here they are. So first I want to have us look at this tree over here. And you can see, hopefully, I know the text is a little small, all of these samples are from the northern portion of the range. And they're all clustering pretty well with samples that were collected in the same place. So like all of these are Tucky Mountain, all of these are Cottonwood Mountains. And then way down here at the bottom, nested within all of those samples collected from the north, are all of my samples from the southern population that were included in this first run. And so they're separating out as their own thing, but they are deeply embedded within all the northern population. So that is interesting, and I'm super excited to get more data to put into this tree to parse out those relationships more. And then, but yeah, so geographic signal, yay. I'll have hopefully more results for you soon. And then if we look at this super colorful block here, this is a structure plot. And this is trying to make like an estimate of ancestral populations for the species. And so each color represents a different like number of ancestral populations. Hope I'm explaining that correctly. Okay, and so my best K value was K equals 4. And basically the takeaway from this plot is that there's definitely substructure within each of these populations. And so that can have important implications for any type of ex situ conservation that may be attempted for this species in the future. And yeah, again, not very much data, so I'm excited to have more to see if those patterns hold or not. And I still have a year left, and so first and foremost, I need to finish my field work like right away in the spring, and then finish making my species distribution model. I actually made a super preliminary model like last week, and so I'm excited to refine that more very soon. finish my population genomics analyses once I have all my data back. And then once I have all that information, I will make conservation recommendations and update those metrics of rarity that we talked about earlier. And hopefully make some conservation seed collections, because this species is not currently seed banked, and it would be really awesome if it was. But it's hard. And finally, I would just like to thank everyone who's helped me in the field and in the lab, and the generous support of all of my funding sources. And with that, I'm happy to take any questions that you might have. Yes, who wants to go first?
It looks like in your structure plot and phylogeny that there is like a latitudinal signal.
Yes.
Do you have any thoughts about that? And do you have any thoughts about, what, yeah, I don't know, just like the southern population and its occurrence, and like, obviously it's hard to make inferences just based on this, but do you think it seems closely related to the most southern?
Yeah.
I don't know, what are your thoughts?
It is coming out as most closely related to the southernmost northern populations, which is interesting. And I'm thinking about what that means now. Somebody brought up budding speciation to me at Botany when they saw this on my poster, and so that's something that I'm exploring, and also like relic populations, and reasons why that might be. So yeah, I'm super excited to have more data to maybe parse that out a little more. Yeah. Yes.
Sorry. Did you have someone else before me?
Chloe was raising.
Oh, sorry.
Oh, I was just wondering, what, can you just remind us what your outgroups were?
Oh, yes. So I have all three other species in Salvia, subgenus Autobertia, subgenus Spacey, which is Salvia cartuacea, Salvia gradiae, and then Salvia californica.
Okay.
And then Salvia mellifera, which is in the sister section as like my outgroup. Yeah.
Oh, now I have two
Okay.
questions.
Sorry.
One, bouncing on Chloe's, did you do like herbariums material for like the bog plant?
Yes.
How did that do?
Really well.
Oh,
Yeah,
that was
that one, it extracted really well, it sequenced really well, so. Yeah, that was the only one that I did, I sampled herbarium material for, but I worked.
I'm gonna pick your brain about that later.
Okay,
And two, when is your field work in spring?
probably like the very beginning of March
Okay.
is when I'll try and knock it out.
I might pencil myself in.
Okay,
Okay.
sounds good. Mahima was my first field assistant for this project, so. We had a good time. Yeah. Yes.
What do you think Salvia funerea smells like?
Oh, it's like, I love it, and my description is not gonna make it sound good, but it's like a little sweet oniony, but like mixed with a little desert musk in the best way.
Yeah.
Yeah, that's my description. Yeah. And anyone else? Thank you so much for listening. It is my pleasure to introduce our second speaker of the day, Garrett Goodrich. I had to write notes. Garrett is a third-year master's student here at CalBG, conducting a floristic study in Sequoia National Forest in the Sierra Nevada mountains. But to me, Garrett is so much more. They're my dear friend, office mate, designated email proofreader, and the person in this room who I have spent the most time in a car with. The summer before I started this program, Garrett did a crazy thing and invited me to help them with field work for a whole week after meeting me one time. To me, at least, it became clear very quickly that Garrett and I were going to get along very well, and I think the rest is history. Garrett's enthusiasm for botany and thirst for knowledge is inspiring and contagious. They ask the best questions and always have a beautiful, nuanced, insightful response to questions that they are asked. And I just think they're really cool. Garrett, I'm so very grateful that I get to do grad school with you. And so, without further ado, please welcome Garrett Goodrich.
That was so sweet. Thank you. To put you on the spot a little bit, to tell you an anecdote about that week-long trip before you had even started. Couple days in, I'd gotten in the habit of being like, what would you like to eat for lunch today? Where do you want to go? Here are some options for field work. And Mia had a little bit of a breakthrough and said, you know, Garrett, I really just don't want to make decisions. And from that point on, I was just like, you know, here's what we're doing today. Here's what we're eating. And that was a breakthrough of sorts. The rest of the week went so smoothly with no more decisions for Mia.
Thank you.
Of course, anytime. So today I'll be chatting about my Flora, now titled Big Meadows and Giant Sequoias, a vascular flora in the vicinity of Boulder Creek, Fresno and Tulare counties, California. So hopefully it'll let me know. What did we do last?
I don't think anything. I think it was on the same screen.
Cool, we're working again.
Okay.
So I want to start by introducing y'all to the study area where I'm working. It's about 75 square miles up in Sequoia National Forest entirely, but it's closest to Sequoia and Kings Canyon National Parks. The southern border is shared with Sequoia National Park, and then the eastern border is Kings Canyon National Park. If you can think about where Fresno is on a map, I'm basically 100 kilometers straight east into the mountains from Fresno. Some main places of note, there's State Route 180, which takes you into Kings Canyon, Hume Lake on one side, and then General's Highway. If you've ever driven that through Sequoia National Park, you've seen my study site, at least in passing. The northern half of my study site, which is in Fresno County, is a story, or a land story, a natural history that can be told mostly through fire at this point. Almost all of this has burnt. There are giant sequoia groves which rely on fire, but the intensity of fire in recent years has deteriorated many of the forested communities. And so there are some areas like you see on the top left, which is the understory of a giant sequoia grove, which is just a carpet of seedlings. And there are areas like you see on the top right, which were once forested and are now mostly non-native grasses and ceanothus, and we can have some hope that they will return to forested communities, but I'm not so sure that the fate of that system is clear at this point. The southern part of my study site in Tulare County is all about water. And so there's Big Meadows, a named locality right here, which you can see on the bottom left, a huge meadow system of many different meadows with a river running through it. And then the Jenny Lakes Wilderness, which is the southernmost part of my study site, is a set of high elevation lakes in the Sierra, a photo of which Weaver Lake is in the bottom right for you. What really drew me to this area is these two pockets of noted biodiversity. These are two areas that have been flagged historically by botanists as kind of centers of rare plants in the area. This one in the north is associated with a limestone outcrop, and then Big Meadows itself seems to have some really interesting, rare, and fun plants. Talking about the study site and the plants you'll experience there a little differently, let's look at elevation. So my lowest elevation is about 3,500 feet, but there's just a little bit of that. It's right by the Kings River, just a short corner of my study site, and you quickly gain elevation and move out of chaparral into yellow pine forest, including giant sequoia groves, red fir forest. This is where a lot of my study site is. A lot of this is old plantations from the Forest Service. Some of it is forested community that has kind of built itself up through time. And then at the higher elevations, there's a lot of lodgepole forest, and this is in the southern part of my study site. There's meadows with lodgepoles in and around them. This is where you'll find the lakes, and I get over 10,000 feet in elevation at my highest point. Interestingly, it is lodgepole straight to the ridge in my study site. In adjacent parts of the Sierra at that elevation, you might hope to see some of the other fun high elevation tree species. Not here. It's lodgepole all the way. So to talk about my research a little bit differently, now that you know the study site, I wanted to test these two broad hypotheses. First, that there's still undocumented floristic diversity in that study area. And I formed this hypothesis through searches of historical specimen data. This looked like a gap relative to everything around it. And then I wanted to ask questions about how anthropogenic actions might impact the plants that grow there, especially the rare plants. How am I going to test that? I'm going to collect plants like mad, identify them, and then deposit those specimens to herbaria. And then out comes over 1,000 herbarium vouchers, looking at historical specimens to make an annotated species checklist, and then improved rare plant mapping in the study site, which hopefully would be shared back with the Forest Service for improved management. So to give you a summary and a nice pretty map, this is where collections stood before my study. You'll see that there's some really good work in the Jenny Lakes Wilderness that was done by people from CalBG. And there's a lot of effort, I'll let you guess where the road was, or is still, straight through the center along Big Meadows Road. And then scattered collections elsewhere. There's decently good roads in the area. This is what I was able to accomplish in my first field season. So recovering ground that had been seen before, and filling in some of those gaps. You'll notice that I didn't make any collections in the Jenny Lakes Wilderness. I didn't have permits for this field season to get into the wilderness. But fortunately, this year with my fieldwork, I was able to get into the wilderness. And you'll notice that I filled in a few more of those gaps. This is actually collections as of the end of July. And so there's better sampling in the Jenny Lakes Wilderness, particularly the left half of it now. And some of these gaps that you see on the map, particularly this one in the center at the northern part of my study site, will very likely remain a true gap. It's Boulder Creek, which is a deep canyon that's cut by Boulder Creek and is incredibly inaccessible. It always has been, and since the 2015 rough fire, any trail that got near it is no longer maintained. So I've tried from just about every access point you could imagine to get in there, and this is where things stand. To give you some numbers, I'm up over 1,000 collections now, which is good because I'm almost done. These were made over 93 days of collecting, 23 trips, and I've had the help of 13 field assistants so far. People often wonder, like, what does it look like to collect, or what do you do about the weird plants? What do you do about the poisonous plants? So I tossed in this photo of me collecting a plant that does cause severe skin irritation if you touch it. We as botanists have all kinds of tools we use to get around these problems, but ask one of us how to collect a cactus or an aquatic plant sometime. I think it'll end a fun story. Of the things I've collected, I've identified 17 of them as rare so far. Number subject to change, hopefully will rise as I keep identifying. Of those 17, 8 of them had cattle grazing documented either on the rare species or in the immediate vicinity. And then logging activity was documented around 11 of those 17 rare taxa. So to talk about the threats a little more clearly, I wanted to talk about logging first. This is a photo of a logging camp right adjacent to a giant sequoia grove from 1913. This is the Hume Bennett Lumber Company. I think that's the name of it. It changed through the years. They had railroad tracks through my study site, logging camp set up. This is, I believe, Camp 7, which implies at least 6 more. All of the railroad tracks and camps are gone. Anything that was left burnt down in 2015, but the railroad grades are still something that I use to access my study site and exist today. And logging this was mostly clear cutting of giant sequoia groves at that time. The face of logging has changed through the years. Now, most of it is done as fuels reduction or hazard tree removal near roads. But this is what that looks like, this is a photo that I took in my study site along General's Highway in 2024. There's still massive understory disturbance when they're doing fuels reduction and hazard tree removal. Talking about fire and cattle grazing, two other threats. I keep talking about this rough fire. It was one of the catastrophic wildfires of its time, although it's been pushed down the list as time goes on. This top half of my study site is the rough fire burn scar. And it comes out to about 50% of my study site has burnt since 2015. This is a photo of a hotspot that I saw while I was in the field. Fortunately, this went out. This was in 2024, didn't become big wildfire, but it really underscores the threat of fire in the study site, that they keep starting, many of them go out, some of them go on to have catastrophic fates. Cattle grazing is also something I think about a lot. Past a certain point in July each season, my meadows are no longer able to be collected. They've been mowed down like lawn by cattle. Yeah, there's arguments for the role of fire and cattle grazing in these communities. But the impact that they're having here is certainly at least sometimes negative. And then talking about climate change. So I have the 75-year averages for you before my study started, and then the graphs are specifically showing how far each year for those 75 years was from that average. On the left, we have temperature, and so you'll see a really strong warming trend. Most of the years that are warmer than average have occurred in the second half of that 75-year span. And there's a similar trend of drying for precipitation, although less clear. These are modeled data for just a central point in the middle of my study site. The weather station data from my study site exhibits similar trends. I just didn't want to show you a bunch of graphs, and I wanted to give you one tiny one. So let's talk about those rare plants that I've come to love so much. How we talk about rarity in California is mostly shaped by the California Native Plant Society, who has this wonderful system for us. Every rare plant gets a code after it that's letters, numbers, a period, and then another number. What that means, a 1B is rare or endangered, either entirely in California is where it grows, or maybe some of them grow a little bit outside of California. 2B is rare or endangered in California, but more common elsewhere, so in a different state, usually. 3 is the needs review list, and 4 is, it deserves some rank, but maybe it's not a really rare and threatened species quite yet. And then after the decimal point is the threat rank. So a 1 meaning that it's seriously threatened where it occurs, and a 3 being it's not very threatened. So here are some of those rare plants from that northern part of my study site. There's this lovely Eriastrum, which I've had some help from an expert identifying recently, that occurs up right by this limestone, although not on it. And that was a new find for my checklist, so I'm very excited about that one. There's also this I find it cute, Ceanothus. It's the Fresno mat that grows, again, not on, but very near the limestone up in this hotspot. There are about 10 individuals growing. As far as I can tell, that number has held steady since at least the 90s. And I'm very interested to go back and look at this the next time I go out, or maybe early next season, or the next time I can get myself there. Its entire habitat is in or immediately adjacent to one of those old railroad grades, and they're doing hazard tree removal on that road right now. And so I'm curious if these plants will persist past this logging effort this year. Let's talk about big meadows. This is a lot of fun. This is where the water is, and so you get kind of showier plants sometimes. This is a carnivorous aquatic plant that's rare in the state of California. This is just hundreds of flowering stems, and in the center of big meadows, there was just pool after pool like this. It's incredibly abundant. Similarly, this Dicentra and this Ivesea, either Ivesea or former Ivesea, I can't remember right now, both grow as roadside weeds around big meadows, and they're rare plants in the sense of the state of California, but they are incredibly abundant in my study site around Big Meadows. And then I've added some rare stuff this year, two that I especially like. I was able to relocate and then add some new populations or occurrences rather of Fritillaria pinetorum. And then maybe the most exciting plant that I've found in my flora is this Lamiaceae collagii subspecies collagii, which is a very little cute thing. But this is now the southernmost place that an herbarium specimen has been created for it. And before I created this voucher, the next nearest one, the southern one, was 50 miles north. So this represents, at least in herbarium records, a 50-mile range extension. Floras are not just documenting this stuff, they're also using that information to do something. And so the conservation contributions and collaborations that I've been able to make over the course of my project have been such a joy. I'll flag just a couple here for you. One about Eriogonum nudum. Charles, who's here, is working on that for his PhD, which is so fortunate because I encountered in the field this population that is entirely problematic. It kind of looks like variety regiripum, which would be rare and significant if it was that. It also kind of looks like two different varieties, and you can key it out each way depending on the specific stem you're looking at. So Charles was in my study site with me about a month ago and grabbed collections of all of these different forms that occur together, and we'll hopefully get genetic sequence on that and clear up some of those problems. iNaturalist has been a great tool and something I'm so grateful to be able to contribute to over the course of my study, especially for stuff that I find that's rare, that is outside of my study site, where I don't need to make a voucher, probably shouldn't make a voucher, but I do want to document it when I'm going from point A to point B. This being a Lamiaceae Bramsii example of exactly that. Of course, I'm combing through lots of databases for the historical data, which inevitably raises problems. This Caloptridium pygmaeum hasn't been seen on the western side of the Sierra in decades at this point, I believe it is. There was a polygon in one database suggesting that it should occur in my study site, has historically occurred. When I was digging into it after searching for it for hours, I realized that it was an error, and it was actually a record from a different big meadow in Tulare County. And so this is one example of an error in a database that I've been able to kind of clean up and help along over the course of my study. And then finally, I'm working with the Forest Service as much as I can to flag these concerns of land management. So this is that same area of logging that I sent you pictures, or I showed you pictures of, sorry, before. The piles of logs that you see in the foreground are actually toppling back into a meadow where Ivesea campestris and Osacchia longifolia variety cuprea grow. And I documented those just a couple months before they came through and squashed those individuals with logs. So next steps, where do I go from here? I need to finish my field work. I have one more trip that I am hopeful I'll get to go on, although there's a fire in the region right now, so maybe smoke will prevent that. And if I do get to go, I would love to see that ceanothus again, see how things stand with it, and then target the high elevation Jenny Lakes wilderness, grab some aquatic plants, some lake meadow things, and then that could be it for field work. I need to key out all of my specimens or everything that's left unkeyed. So that's going to be a big chunk of my time going forward. Annotate historical specimens to build out that annotated species checklist, write up my thesis, and then publish it. And I have these wonderful illustrations from Emily Franz, who's here with us today, that will go into my published flora and help to kind of extend, once again, the outreach potential of this project. With that, I'll say thank you especially to everyone who has helped me with this and is here today. It wouldn't have been possible without y'all. Of course, thank you to my funding sources, and I'm happy to take any questions.
How did you choose a specific area?
Yeah, a good question. It was very iterative. Started with just kind of big searches on CCH2, which is the database for herbarium specimen records, to just try to find gaps because I wanted something that was relatively under collected where collections would have more of an impact, hopefully. And then I visited a couple areas. I was kind of pursuing multiple leads on interesting looking areas at the same time. And I don't know, nothing quite was sitting right. So I made one last trip to a potential place that I had flagged, which was my study site. And I remember driving up the mountain, going through all of the communities from the Central Valley up to the giant sequoia groves in one day and just being floored and knowing like this, I want trees, I want water, I want rare plants that have some need for love. And it checked all those boxes. Yeah.
You gave the example of the Ariagon of Newdom. Do you have any other taxa for which you were just either totally stumped or just like banging your head against the herbarium counter? And then also, do you have a prediction for what will be the most species-rich genus?
Oh, two good questions. Yes, there are specimens that I, at least at this point, there's so much keen to do. I just put them back in their folder. I'll see you later. So I don't know who they are yet, but they're swooping around. I mean, Carex is low-hanging fruit here for a guess with all of my water. So I'm gonna say that one preliminarily, probably Carex, if I did it right. Yeah.
Besides fire being a sort of direct threat, are you seeing a lot of tree mortality or is that an issue?
It's hard to tell. A lot of the non-lodgepole communities are plantations, like I mentioned. And so it's kind of hard to know, they're like even age stands. I don't know what's going on with them. It's hard for me to read. And then lodgepole is all I have at higher elevation, which is just incredibly resilient. And it can take a beating. And so I haven't noticed tree mortality outside of fire. And that might be a dual effect of land use history and fire being such a big mark on the landscape.
Another question following up on that. How confident are you really that it's only lodgepole all the way up? Like,
I know, it drives me crazy. My dad is here and can talk about me being like, okay, just sit at the base of this hill while I hike up this scree for a couple hours and I just look at every single tree. And like from a distance, I'll be like, that's a little too twisty, or you foxtail, and I hike to it and I'm like, oh, like of course you aren't. So I've spent a couple field days at my highest elevation places, just pulling my hair out, being like, it's another weird lodgepole, or oh, it's something else that looks weird because it's too high. But I feel pretty good about it. Travis has noted it and Leroy have noted it, and they're both botanists that I trust to kind of flag when the trees are being a little weird.
Oh, I was gonna say, you've been working with the Forest Service to the extent possible.
Yeah.
For an example, like you gave, how receptive have they been, and like, how has that communication happened, and?
Yeah. Part of this gets bogged down in Sequoia National Forest management. The first season I had a much easier communication channel because they had a staff botanist. And so I was able to reach out to her and talk to her. She left at the end of my first field season. And since then I have not had any luck getting in touch with anyone who has any appetite for hearing this. And even when I did have communication with her, it was really hard for her to get the other people motivated to respond. So it's been really slow going.
What was like your single like favorite find like out in the field? Like what plant got you the most stoked when you came across it?
I think I'll go back to that. Lamiaceae, which I have more story about it. I was like at the place where I usually camp, brushing my teeth in the dark, and I like saw what looked like a piece of like a grocery bag, just like white fluff stuffed to the ground, and I was like, who's littering in this campsite? And so I go and I pull it and I was like, oh, this is a flower that I don't know. And so I went back the next morning when there was light and the flowers were open and I was like, this is a flower that I really don't know.
Yeah.
And so I took lots of pictures and it was keying out to this Lamiaceae and I was like, wait. This is a flower that shouldn't be here and I really don't know. And they were like vanished almost instantly. They flowered for a week or two. I got 3 collections of it and then gone. It was a lot of fun to work with that one.
Did you see any visitors on the fiddle area, kind of fun, like insect visitors? It's
No.
such a cool color.
It smells horrible.
I don't know, it's like, wonder if it's like fly.
It's fly, I'm gonna ask, yeah.
Yeah, I
It
hadn't
smells
noticed.
like rotting.
It's so brown.
Yeah.
Brownish red.
Neat.
I usually caught them a little early or a little late. Like I have lots of vouchers of them where all of the stems, because they were abundant where they occurred, are in fruit, except for the one flower that I was able to take. So I didn't have a lot of interaction with like peak flowering.
Yeah.
It's interesting to know that they smell bad.
They just look like big old flies with of it.
Yeah.
I've got pictures of
Oh, cool.
Oh, to see those. Okay. Thank y'all.